Okay, so we've got a few more folks joining us, but I wanted to go ahead and introduce Anson Blanks, who's going to be presenting the data today. He's a customer success scientist at Phytonics. I'm Sutton Thomas, research application scientist at Phytonics. So if you're new to our webinars, we really try to keep them interactive. So please throw out your questions and I'll interrupt Anson during this. But just to give Anson a brief introduction in terms of his background, he's a registered clinical exercise physiologist. He earned his PhD from Virginia Commonwealth University in 2018, and most recently completed his postdoctoral training at Augusta University in the Laboratory of Integrative Vascular and Exercise Physiology. And in keeping with the theme of 2020, Anson enjoys craft beer, quiet rooms, and drinking craft beer in quiet rooms, which is actually fully compatible with what's been happening this year. So with that, just wanted to introduce Anson and let him present the new data that he has using NovaFluors in a conventional panel.
Thanks, Ed. I was social distancing before it was cool. So thank you to everybody that's joining us here today. So we're going to be talking about flow cytometry panel design using NovaFluors and how to really unlock the full potential of your cytometer and get the most out of your experiment. And as said, please interact as much as you can. So ask questions throughout, and we'll be answering questions throughout the presentation, as well as at the end of the presentation. And if you have any questions, feel free to email either Seddon or I, or info@phytonics.com. And of course, go and visit our website, phytonics.com. If you enjoy flow cytometry at all, we have got a lot of stuff on there for you, a lot of cool information and fun stuff, so check it out. So to start off, with these individuals, they are all very different, but they do have a common theme. So Masaki Watanabe was the inventor of arthroscopic surgery. The Golden State Warriors, pretty recent 3-time NBA champions, and Larry David, who is the co-creator of Seinfeld and Curb Your Enthusiasm. So at the surface, these may seem very unrelated, but the thing that they have in common is that they are all game changers. So they broke from the traditional convention and they really changed their fields. So Dr. Watanabe invented a pretty much almost non-invasive form of surgery that's used globally. The Golden State Warriors really relied on analytics and the three-point shot and completely changed the NBA. And if you've ever seen Seinfeld or Curb Your Enthusiasm, you know that they are very different from most comedies that are on TV. So absolute game changers. With that being said, how can we change the game in the field of flow cytometry? So at Phytonics, we thought about being a game changer by engineering a better panel. So to engineer that better panel, we took superior optical performing fluorescent labels, combined them with panel design to achieve the highest data resolution, which enables us to use panels that enable more markers and higher resolution with no instrument changes. So that's the key. You can use whatever cytometer that is in your lab and really get the most out of it. And I'll show you how to do that. So I'll show you how to take this 11-color memory T-cell panel and expand it out to a 13-color panel without making any type of hardware changes. And with that panel expansion, that's going to allow us to dive deeper into the biology of our cells. So we can really look at these cell phenotypes in these subsets, in this case, CXCR4 and CD28 expression on naive T cells, central and effector memory T cells. And again, that's with no hardware changes. It's just thinking about dyes a little differently. So when we think about panel design, we have to take into account the impact of dye performance on biological resolution that is going to impact our panel design. So the first thing that we tend to think about when designing a panel is going to be spectral overlap. So ideally, you want to space your floors out far enough apart that you really don't have to worry about spectral overlap. But of course, that's almost never possible. So in order to account for that spectral overlap, we perform compensation. The second thing we have to think about is going to be antigen density. And with antigen density, the goal is to match the brightest fluors to the dimmest antigens and vice versa. And our third aspect is going to be spillover spreading. So as we add more fluorophores into a multicolor panel, there's going to be more spillover spreading that is going to be more difficult to account for, and it's going to cause us to lose resolution in our panel and maybe lose some of the minute details that we're really looking for. And then the fourth aspect is going to be the staining index, which is going to be the separation between your positive and negative populations that really allow you to separate those populations and get the most out of your panel.
Anson, we've already got a question. So I know you'll be getting into this later, but just since you were just talking about separation index and staining, we've got a question in terms of what is the difference between 70S and 120S.
Oh yeah, so we're definitely going to get into that later on, but basically it's the floor brightness. So if you think about the wattage on a light bulb, that's pretty much the same thing that we're doing with our S designation. So the higher the S, so the 40S or the 70S is going to be about 2.5 times brighter than the 40S version. So it's going to have a higher separation index.
Great.
So when we're talking about legacy, or when I'm talking about dye performance, today I'm going to be talking about legacy dye performance. And when I'm talking about legacy dye performance, I'm just talking about the traditional dyes that you've used for years in your flow panels. So APC, PE Dazzle, all the other tandem dyes that you typically use. So whenever I say legacy dyes, that's all I'm referring to. With these legacy dyes, there tend to be two major issues that we have to account for when designing a panel. The first is going to be cross-laser excitation or cross-excitation. And that's simply whenever a floor is excited by multiple lasers. So I won't have mouse issues. So for example, with PE and PE Dazzle, we know that their primary excitation is going to be the yellow-green laser. However, they're also strongly excited by the blue laser. Similar problems with per CP. So we're going to have excitation on the blue laser and the yellow-green, as well as the violet laser. And the problem with this cross-laser excitation is going to be that it's going to eliminate a detector. So over here, we've got an example of PE Dazzle that's excited by both the blue and yellow-green lasers. So we're going to have a strong positive signal in both of these detectors. So whenever we're performing a multicolor panel, if we're using PE Dazzle, we can't use both of these detectors at the same time. We've got to pick one. So we're already kind of losing the value of our cytometer. The second aspect of legacy dye performance that we have to take into account is the emission spectrum. So a lot of these legacy dyes have a very broad emission spectrum. In this case, Brilliant 510 is going to have a broad emission across the spectrum. And so one of the ways that we attempt to deal with this is with compensation. So ideally, whenever you perform compensation, it's going to look something like this. So we're going to have APC size 7 and PE size 7 after compensation. Ideally, this is the signal that we're going to get, or the lack of signal. However, in reality, whenever we compensate these dyes, we end up with this fishtail or trumpet effect, which is going to be APC size 7 spreading into the PE size 7 detector. So kind of an easy way to think about that is to think about a perpetrator and a victim. So in this case, the APC size 7 is going to be the perpetrator that's spilling over and spreading into the PE size 7 where it's not supposed to be. So our PE size 7 is being victimized by APC size 7. And whenever we're designing a panel, we have to deal with spread, and an easy way to kind of visualize and conceptualize that spillover spread is going to be looking at the spillover spread matrix that's generated by FlowJo whenever we perform compensation. So essentially, we can use the row sums from that spillover spread matrix to identify fluorophores that cause a large amount of spillover and cause us to lose resolution within our multicolor panel. Once we identify those fluors, then we can develop a plan to really improve them and improve our panel. So whenever we're designing a panel, and this is just an example of panel design on a Thermo Fisher Attune NXT, which has 14 lasers, or 4 lasers, and 14 detectors. And these are the recommended fluorophores for each of the detectors. And if we were to use this 14 detector cytometer and we tried to put these 3 violet fluors in VO1, 2, and 3, because of the bunching and the large amount of spread from the violet fluors, whenever we stack these violet detectors, we're essentially going to have so much spillover or spread into the VO4 that it's no longer going to be a usable detector. So already we've gone from 14 detectors down to 13 detectors. At the same time, if you've ever used any panel design software and you've tried to put PE Dazzle or PE Size 5.5 off of your yellow detectors, you probably get this nice error message that pops up telling you that, yeah, you can do that, but now you can't use your blue detectors. So now our 14 laser cytometer is down to 11 useful channels. Even if we juggle this around and use PE Size 5.5, we get the same thing. So we can't use these yellow and blue detectors at the same time because of the cross excitation that occurs between these, that occurs on these fluors. And this is a similar problem that Fortessa users tend to think that they don't have because of the trigon setup. Really, that problem is still occurring, but because of the trigon, it's simply that the instrument is engineered around the problem. So it's not eliminating the problem, it's just designing an instrument to try and get around it, but it's still there, and that's because of the inherent fluor performance. So using our now 11 color cytometer, we decided to build a memory T-cell panel using the colors and antigens that you see here. And results-wise, pretty decent. So we were able to pull out our our T-cell populations, including our CD4 positive cells, CD8 positive, double negative, as well as our NK cells. So you're probably thinking, okay, you got the results that you wanted. Why should I change any of my fluors? I'm getting what I want to get out of my cytometer. We've got fluors that we've been using for years and years. Why do I need to change? And hopefully I can convince you of why by the end of this presentation. But if you make that change, you can really improve your data analysis by improving resolution. So if we can reduce this spillover spread error, it's going to give us more resolution. So we can see here that the spillover spread that's occurring from PE Dazzle is really starting to spread over into CD56 or into our CD56 detector, which is probably going to cause us to lose some of the resolution of this CD56 dim population. Whereas if we can reduce that spread, we're going to get a more accurate assessment of these CD56 dim cells. So resolution is extremely important. In this case, when you look at this picture, you can probably tell that it's a picture of a bunch of bagels, right? And personally, I could go for a bagel right now. But with 20% resolution loss, it does become a little bit fuzzy, and it's maybe a little questionable as to whether or not they're all bagels. And it turns out we do have some bagels, but we also have some puppies that are just trying to take a nap. So resolution is very important. When we're thinking about a multicolor panel, as we add in more colors, we tend to lose more and more resolution. So in this case, we're getting up to a 60% resolution loss. So you can maybe see that there are some puppies staring back at us, but also some of them are just blueberry muffins staring back at us. And as we continue to build out a larger and larger panel, we're going to lose more and more resolution until things become completely useless, just like this picture. So everything in this picture is pretty much useless. So it's probably pretty difficult to tell what's going on here. However, if we really clean things up, we can see that the whole picture is just a bunch of chicken skin. So given the problems with our legacy fluors, what if you could design your own fluorescent label? And what impact would that have on panel design? So if you were designing your own label, you would probably want it to meet 3 major requirements. You'd want it to be bright and clean, so as little cross excitation as possible. consistent and stable, and it'd be really nice if it had controllable brightness. If you could choose your brightness based on antigen density and your panel needs. And that's exactly what we did with the Phyton structure at Photonics. So our Phyton structure is a synthetic DNA nanostructure, and we've embedded fluorophores into the Phyton structure, which allows us to control the fluorescent properties. So because of this structure, we now have far less cross-excitation and far less spillover spreading in comparison to your legacy fluors. And an added benefit of this Phyton structure is it allows us to control the brightness of our fluor. So we can add more fluorophores onto the Phyton, which allows us to take a fluor that will fit into our detector and expand that brightness based on antigen density and your panel needs. Our excitation and emission spectra for all of our NovaFluors are publicly available on FPBase, as well as Fluorofinder and Cytec.com. And of course, you can visit Photonics.com to get more information on all of our NovaFluors. So the easiest way to talk about improving your panel and talk about the utility of NovaFluors is going to be to talk about NovaFluors and their comparators. So these are some of the comparators for NovaFluors, and essentially you can use the NovaFluors listed over on the right to replace any of these suggested fluors that may be in your panel. And this is going to give you a lot of benefit because you could use NovaBlue 610 to replace PE off of the blue laser, or you could use NovaYellow 610 to replace PE off of the yellow laser. And that's going to provide you a lot of benefit because we know PE has some cross-excitation and also creates a lot of spread. So if we can get rid of that, there's going to be a lot of benefit there. At the same time, if you're trying to use PerCP and PerCP Sci 5.5, you can theoretically use it alongside PE Sci 5 and PE Sci 5.5, but because of the cross-excitation and large amount of spread, using these fluors together is pretty difficult. Whereas if you were to just switch out to NovaBlue 660 and NovaYellow 660, those problems would be eliminated. So just a quick reminder of the results from our legacy panel. And I do want to highlight that all of the fluors in red are going to be the fluors that we're going to replace throughout this panel design process. So these are places that we can make improvements. So when we look at the spillover spread matrix from this legacy panel, this is what we see. So no surprise, our largest spreaders are going to be in the red detector. So we're getting a lot of spread from APC and AlexaFluor 700. After that, we've got a large amount of spread that's coming from PE Dazzle. So if we think about the amount of spread that's coming from PE Dazzle and combine that with the fact that we're losing the detector because of the cross-excitation coming from PE Dazzle, I think it's a pretty simple way to go about improving the panel is going to be replacing PE Dazzle. And that's exactly what we did. So we just took PE Dazzle on CD45RA and replaced it with NovaYellow 610. So same detector, same antigen, nothing about the panel changed except for we took PE Dazzle out and put NovaYellow 610 in. And because we don't have that cross-excitation the way that we do with PE Dazzle, that allows us to open up a detector. So now we have this blue detector that was closed off, so BL2 that was initially closed off, we've opened up that detector. So our 11 useful channels have already been expanded to 12 useful channels just by swapping out this fluor. In addition to increasing the number of parameters, replacing PE Dazzle is going to improve our resolution. So if we take a quick look at the SSM, we can see in that YL2 detector, there's a 19% reduction in spillover spread, and there's an overall 4% reduction in the panel. So that means that we've got a 4% improvement in resolution for the panel overall, which if you think back to those pictures, that 4% could be very significant in really getting accurate results. So when we look at the results from replacing PE Dazzle with NovaYellow 610, it's very similar to what we have with our legacy panel. So we're still able to resolve all of the same populations. We've got very similar separation. So again, we're getting our CD4 positive, CD8 negative, and double negative cells, as well as our NK cells, which aren't shown here. So simply replacing PE Dazzle is going to allow us to open up a detector without losing resolution. So we've got similar performance between these fluors, and we've already gone from 11 colors to a theoretical 12 colors. So if we think back to our initial panel design, one of the problems, or one of the reasons that we were losing a detector, is because of per CP Psi 5.5, which is also contributing a lot of spread to our panel. So our next question is, if we replace that, what is going to be the benefit? So in order to remove that spread, or maybe remove that spread, and also open up this BL3 detector, all we did was take CD3 and move it to the YL3 detector using NovaYellow 660. So we eliminated per CP Psi 5.5, added in NovaYellow 660. Other than that, the panel is exactly the same. And that's going to allow us to open up this BL3 detector. So again, we've gone from 11 useful detectors to 12 useful detectors. When we look at the resolution via the spillover spread matrix, you see that adding in the NovaYellow 660 actually does increase the spread a little bit. And that's because we're really just stacking those yellow detectors. So we've got our fluors that are a lot closer together. And also NovaYellow 610, it tends to be quite a bit brighter than per CP Psi 5.5, and brighter fluors usually have more spread. And if you want to check out the brightness of all of our NovaFluors in comparison to the legacy fluors, you can go to phytonics.com. We have the separation indices for pretty much all of the fluors out there, and definitely all of the NovaFluors. So I believe the separation indices or index for NovaYellow 660 is about twice as much as it is for per CP Psi 5.5. But even with this overall increase in the spillover spread matrix, we still have an open detector, which in my opinion is worth having a little bit of resolution loss, which that resolution loss doesn't have a whole lot of impact on our panel. So even with the increase in the SSM, we're still able to pull out all of our populations of interest. So we've got our CD4, CD8, double negative, and NK cells, which again aren't shown. And when we look at the performance of NovaYellow 660, we're still able to pull out our populations. So one of the big benefits of replacing that per CP Psi 5.5 is obviously opening up a detector. But in addition to that, you also get to get rid of a tandem dye, which I want to start off by saying that NovaFluors are not tandem dyes. And that's very important because tandem dyes tend to have a lot of issues. So one of the big issues with tandems is going to be their instability in solution. So this is an example of APC Size 7 as well as PE Size 7 that were in solution that have started to degrade into their individual dyes. So APC is degrading into APC and size 7. PE is degrading into PE and size 7. And that's going to cause a false positive signal in the APC detector and a false positive signal in the PE detector.
And so I'm going to interrupt you real quick. I think it might be helpful to explain, why we would say they're not tandem dyes if, there's a donor molecule and an acceptor. So part of that is, yeah, go ahead.
So it's basically down to the actual Phyton structure. So because the fluors are on the same Phyton structure, we're not going to have the degradation problems that we tend to have with tandem dyes.
Right. So tandems, often say a PE size 7, that size 7 is actually attached to the PE, whereas these are actually fixed on the fluor at a specific amount. So we don't have the same issues in terms of tandems, that there is that reproducibility and not the degradation.
Right. And along those same lines, the problem that we're not going to have with NovaFluors that we tend to have with tandem dyes is going to be a differential decoupling based on cell phenotype. So we can see here on lymphocytes, the decoupling of this APC size 7 is at a much slower rate than it is on monocytes. Furthermore, tandem dyes do not like fixatives. So this was APC size 7 that was in 2% paraformaldehyde. So when analyzed at 0 hours, we get the signal that we would expect. So a strong APC size 7 signal, a negative APC signal. However, after 72 hours, as the APC size 7 degrades into the individual APC and size 7 molecules, we start to get a strong false positive signal in the APC channel. And probably one of the most annoying things about tandem dyes is going to be their significant lot-to-lot variation. So this is going to make performing repeat experiments pretty difficult, because when using tandems, you want to use the exact same lot so you have some form of consistency. So this is the same antigen on the same color tandem, so APC size 7, just from different lots, and we can see how much variation there is between those lots. So if you're doing compensation or multiple experiments that you want to compare, you've got to use the same color, same antigen from the same lot when you're dealing with tandem dyes. And all of that can be quite a pain.
Anson, we have another question. What about the BuV dyes, which are polymers? Do they also have the tendency to degrade?
So you'll probably want to follow up on this one, Seddon, but I don't think they had the same degradation problems, but I do think that because they are polymer dyes, there is a problem with aggregation. Is that correct?
Yes, there's that, but also basically BuV, beyond BuV 395, it is actually a, functioning as a tandem. So you basically have anything above that in the spectrum. So there's, the donor molecule is coming from the BuV 395, and then there's the acceptor. So it's less about the degradation and more about the lot-to-lot stability. But there is, some process happening there, and those have to be made under pretty specific conditions, as well as the, the issues that happen when you actually start to use them together and have some exchange. And, that's why you have to use the brilliant buffer. So thanks for the great question, though.
Yeah, that's a really good question. All right, so I've shown you how you can basically upgrade your panel from 11 colors to 13 just by replacing the PerCP Sci 5.5 as well as the PE Dazzle. And that's going to allow us to open up these blue detectors. So now we've got BL2 and BL3 that are open, so we can move from 11 colors to 13. However, you probably guessed that you can't just throw in our traditional tandem legacy dyes, or we're going to have the same problems that we had before. But thankfully, we can drop in NovaBlue 610 and NovaBlue 660 into these detectors. And for this panel, we looked at CXCR4 and CD28, but of course, you can put a NovaFluor on any antigen that is of the interest to you. So whatever you want to look at, we can put it on a NovaFluor and improve your panel. So we've gone from 11 colors to 13. And one of the big aspects of expanding this panel is going to be the additional antigens that we're looking at, in this case, CXCR4 and CD28. We know that these antigens are fairly lowly expressed, so we need a pretty bright fluor. So we want to match fluor brightness to antigen density. So one of the really nice things about the S designation, which we've already talked about a little bit, and we'll talk about a little bit more, is it allows us to increase fluor brightness without completely rearranging our panel to try and accommodate these dim antigens. So as I was talking about before, with the S designation, we can take this NovaBlue 610, which we know fits into the blue detector, and because we're using it on CXCR4, which is dimly expressed, instead of using the dimmer 30S version, we decided to use the brighter 70S version. Same thing for CD28. Whenever we know it's lowly or not highly expressed, so instead of using the 40S version, we decided to go about 3 times brighter and use the 120S version. And this is just an example of the S versions of NovaBlue 610 and 660 on CD4. So we can see that the 70S is about 2.5 times brighter than 30S, and the 120S is 3 times brighter than the 40S version. So it's a really easy way to match antigen density to fluor brightness, or match fluor brightness to antigen density without completely rebuilding your panel. So we've gone from 11 colors to 13 with the very simple addition of 4 NovaFluors. And when we look at the results from that, this is what we get. And you're probably thinking, hey, this is less data than you were showing before. Why do you have more colors and less data? But in fact, we have so much additional data that I couldn't get it on to one slide. So we've got all the subsets that we had before. So our CD8 positive, double negative, and CD4 positive cells. But now we can really dig into the biology and look at those different phenotypes, specifically CXCR4 and CD28. And we can look at that antigen expression in naive T cells, central memory, and effector memory T cells, as well as our CD8 positive subsets. And of course, you can put these NovaFluors on any antigen of interest to you to look at a deeper phenotype of whatever you're doing your research on. So we've gone from 11 colors to 13. And when we look at the spillover spread matrix of our 13 color panel, this is what it looks like. Doesn't look too bad. Since we're stacking our blue detectors now, we've got a lot of spread coming from NovaBlue 61070S. However, I think this is going to be pretty acceptable considering you're getting a whole new detector in there. But the question becomes, can we improve this panel even more? And you probably guessed that the answer is yes. And we can make that improvement by adding in a couple of NovaFluors. So in this case, if we replace AlexaFluor 488 with NovaBlue 530 and replace PE, which is really going to clean up the panel because we know PE is cross-excited and has a lot of spread. We replace PE with NovaYellow 570. It's probably going to lead to a better panel, which it in fact does, as we see here. So it's pretty much a reduction in spillover spread across the board, but the big thing is going to be the overall reduction. So about a 26% reduction using 6 NovaFluors instead of 4 NovaFluors. So we got a 26% improvement in our resolution, which can be very significant whenever we're trying to analyze those dim and rare populations. So again, the results from our panel look very similar to our 4 NovaFluors. We're still able to resolve all of our populations of interest. We're able to really get into the deeper phenotyping of our T cell subsets. So we're getting all of our information plus a lot more in comparison to the 11 color legacy panel. In addition to that, we are also able to, since we had that improvement in spillover spread matrix, we're going to get better resolution, which is going to enable you to really set your gates a little bit easier and find those populations more accurately and just clean up your data and really see the improvements that you should see whenever you improve your resolution. So essentially, our big point is to get what you paid for. So whether you've got an Attune, a Fortessa, a Cytoflex, it doesn't matter. You can take whatever your cytometer is and put those lonely detectors to work. So I know all of you in your panels, you've probably got a lot of empty detectors that are just sitting there because you can't find a fluor that really fits into your panel. Now you can. Now you can really expand your biology and see what's there. So you can take these subsets that we see here, and instead of stopping your analysis there, you can build it out and really get into the biology. So we can look at all of these subsets and the expression within these subsets. I will give you a warning that whenever you expand your panel, it's going to give you a lot more analysis to perform. So you're going to have to plan accordingly. All right, so I've talked about how NovaFluors are bright and clean. I've talked about controllable brightness, but another requirement for a good label is going to be consistency and stability.
So I'm going to interrupt you real quick. Just we have another question, and some of it we'll be addressing later in the webinar. But what is the cost for getting an antibody conjugated, and is it cheaper to get conjugated to 30S versus 70S? And
So there's no price difference between the 30S and 70S versions. The price of getting an antibody conjugated is going to depend on your method of conjugation. So we have conjugation kits that you can use in your own lab, or you can send your antibodies to us at the 500 microgram scale and up. And I do believe that pricing is going to be based on the quantity of antibody that you want. And was there another part to that question?
So I think, yeah, the only other, I think you covered all the question. The other thing is that any of the antibodies that we currently have in, basically in our catalog can be put on any fluor. So this, basically the wave 2 labels are in process of starting to release in January. So that will, basically be the only deterrent there. But, if you actually already see, an antibody that we have, and Anson will be going into that later, that we can easily put any of the labels on those antibodies.
Yeah, and there's no additional cost. So if you see something on our website, so we have recommendations of colors and antibody combinations, but if you want that antibody on a different color, all you have to do is ask. There is no additional cost. All right, so for our stability, this is Nova, or our Nova Blues and Nova Yellows conjugated to CD4, and the blue line represents 14 days at 4 degrees C, and our orange is going to be 14 days at room temperature. So they just sat on a bench for 14 days. And then we looked at the separation indices over time, and we can see that there is basically nothing to see. So there's no significant change in the separation indices. So it's very stable. Our NovaFluors are very stable, even at room temperature. So if you've got a new graduate student or an absent-minded lab tech, you can take a little bit of worry off of yourself, because you don't have to worry about whether they put the antibodies back up. So again, a big problem with tandem dyes is going to be their instability. But again, NovaFluors are not tandems, so we don't have those stability problems. So this is an example of Nova Yellow 610 conjugated to CD3 that was analyzed fresh and then left in formalin for 2 weeks. And there was no significant change in brightness. So after 2 weeks, still stable, still ready to analyze. So what about consistency and stability? So more specifically, what about your lot-to-lot variation? And if you look at these graphs, so up top we've got our Nova Yellow dyes, and on the bottom our Nova Blue dyes. Each color line actually has 2 lines. So the 2 lines that represent our 2 lots, it's very difficult to see because there is almost no variation. So about the only place that you can really see any type of variation is going to be for our Nova Blue 530 down here. And even this difference is less than 5%. So we've got really, really good lot-to-lot variation, which is going to make repeating your experiments a whole lot easier. So again, how can we change the game with flow cytometry? We can engineer a fluorescent label that has superior optical performance that enables more questions per cell. Add that to a panel that is designed to achieve the highest data resolution, and together those are going to enable us to design a panel that can examine more markers with higher resolution with no instrument changes. So that's the key. You don't need to buy a new cytometer. You just need to make your cytometer do what it's supposed to do. And these are our currently available dyes. So we don't have these split up into our wave 1 or wave 2 dyes, but our wave 2 dyes will be shipping very soon at the first of the year, as said and mentioned. So any of these colors, you can definitely find something that will fit into your panel. We currently don't have UV and violet dyes, but those will be coming very soon. So stay tuned. We're going to have some pretty exciting news with the UV and violet very soon. And as we were discussing before, this is our antibody menu. So these are the antibodies that we have available that are ready to go that we can get to you very quickly. As said and mentioned, you can have these put on any color. If you go to phytonics.com, we do have some recommendations or color recommendations that we have, and that's what we found that have worked best for us. But according to your panel needs, you can have any of these antibodies conjugated to any of our NovaFluors. So whatever fits into your panel. If we don't,
I think they actually work, well on, a number of floors. The reason actually that we have the color designations is that we did some amount of matching the brightness to the antigen density in terms of sort of if you wanted to use, say, all NovaFluors, that's the way they're matched up. Is this would be sort of looking at a more optimal computational panel design. But again, that's why this offers so many options that you can do that. And we did have a question actually specifically about the cost of the already labeled antibodies in the catalog. And I believe for 100 tests, it's is it $395? I was just checking the website myself to double check that I was giving the right answer. We also have 25 test versions of the antibodies. So.
Yep. So, and if we don't have the antibody that you want, we can always conjugate that. So we do have an antibody printing program. So basically you would send us your antibody and we would put it on the color of your choice or the NovaFluor of your choice. Our antibody printing program does start at the 500 microgram scale and goes up from there. So there is an option if we don't have your antibody and you don't want 500 micrograms of antibody, you could use one of our conjugation kits. So our conjugation kits allow you to attach your antibody of interest to any of our NovaFluor colors. And the process for that is pretty simple. So it's a 2-day process, but the actual hands-on time tends to be around 30 minutes, maybe a little bit shorter, a little longer, depending on how quickly you pipette. But just a few pipette steps and then an overnight incubation. And then your conjugated or your NovaFluor conjugated antibody should be ready to use. There's only one precipitation step that's now included in this. So with that being said, I would definitely like to thank our Phytonics team. We really are a team of superheroes. I'm impressed every day that I go into work. It's been a whole lot of fun since I've been there. So thank you to everybody for contributing to this presentation. And you can reach me at ansonblakes@phytonics.com. Sedden's email is up there. And you can also email info@phytonics.com if you want more information. Of course, you can visit phytonics.com, which again has a lot of interesting flow stuff. And with that, I think we'll take some questions.
Yeah. So while we're waiting for some questions to come in, one thing I think that's great to highlight on the website, if you are sort of saying, okay, you've got all these colors, what is best for my panel? And, what we've done is really focus on the wave 1 floors, but now we have, obviously the Nova Reds as well. So you can actually see both the separation index and the spread into other channels is measured on an Aurora. So that gives you some sense of, okay, how is that going to look? And in terms of the naming, again, everything is very clearly named. Nova Blue 530 is, basically just off the blue laser and, at that 530 nanometers.
Yes, so our naming convention is pretty simple, as Seddon was just describing. So the color is going to be the excitation laser and the number is going to be peak emission. So.
I got a comment. Can I unmute? I don't know if
is there something I need to do?
I don't know. Somehow it's like I've joined it twice. So I apologize if that isn't coming through.
Unmute.
Or I don't know if, for discussion. I think that's a good point. I don't know that, I guess they were asking if we could unmute them so that they could discuss is what,
I
was answering the wrong question.
We can do that with the webinar.
I think the default settings on the webinar aren't set up very well for that unless I'm unaware of something. I'm trying to see if there's any settings that I'm missing, only because I've never tried to do that during. Yeah, I can't find that it's an option.
Yeah, I don't think we can do that. I'm looking for it. I don't see the option either.
We'll look into it for the next webinar. I just apologize. I've never actually tried to do that during the process. So, but yeah, thanks so much for the really good questions. If you have any other discussion things, we will continue to read that out. But I just don't think I can actually unmute it. Well, I guess we can hang out a little longer if there's any other discussion questions. But I want to, at this point, thank everyone for coming, and please, let us know if you have any further questions specifically for your panel. I'm happy to look at, your specific cytometer, or, Anson can also answer some questions. But those are things that we can do to help you out as well. So. Well, I guess without anything else, then thanks for coming.
Yeah. Expand your panel.
Yeah.
All right. Thank you, everyone.
Thanks so much.